Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and good day everyone. Okay, so today I'm gonna record this lecture on the chapter 3 part 2 on the metabolism and the reproduction. Okay, so if you still remember, this is the mind map that I designed, and then in the previous part of this chapter, I've shown you this, and we have done the number 1, yes, the taxonomy. We have gone through the taxonomy of the bacteria, and then we've gone through on the technique, the aseptic technique on how to isolate the bacteria. And also once you have successfully isolated the bacteria, we can proceed with the bacterial identification and how to identify them through the most basic steps. The first one, which is the gram staining, to categorize them into whether they are a gram positive or a gram negative bacteria, and also we can classify them through their morphological characteristics. Okay, so for today we're going to continue with number 5, the nutritional needs, and number 6, which is the microbial growth. Okay, let's see if I can change it. Okay, so the microbial growth and the nutrition needs, in general, the basic one, they need a source of carbon, source of nitrogen, and a source of energy. Of course, they will need a lot of things, a lot of other molecules, a lot of other nutrients, but these three are the most essential components they need for the microbial growth. Okay, so what do they need the carbon for? Is for the cellular constituents. What do they need the nitrogen for? Is to make the proteins. And what do they need the ATP? The energy in the form of the ATP is for the cellular proteins. Cellular functions. Oops, sorry. Okay, so there are two nutritional requirements for the growth. The first one is the autotrophs. Okay, so I think you have you are familiar with the name of the autotrophs, and we know what it is by now. So autotroph is the type of microorganisms that can grow using only the carbon dioxide, water, and the inorganic salts. So basically, these type of microorganisms, the autotrophs, they are the producers in the food chain, and because of they are the producers in the food chain, they have to make their own food. Yeah, so how do they make their own food? So usually they make their own food through the photosynthesis. So the outcome of the photosynthesis, not just to give them food, but also to give them energy. One main difference between the autotrophs and the other category is the microorganisms that lie into the autotrophs category, they do not normally cause disease. So they are the good autotrophs. So they are the good bacteria. So normally they do not cause disease. So the second category, based on the nutritional requirement, is the heterotrophic bacteria. So in this case, the heterotrophic, they are more complex than the first one, which is the autotroph. So that's why they need more complex substances for growth. So they require an organic source of carbon and obtain energy by oxidizing or fermenting organic substances. So this is in contrast to the autotroph. The autotroph, they are the producers in the food chain. So they are the first one in the food chain. So that's why they have to make their own food through photosynthesis. But heterotrophic, they need the organic source. So most likely they will eat the producer in the food chain and that is their source of the organic carbon. And the other main difference between the autotrophs and the heterotrophs is that this point. All human bacteria fall in this category. Yeah? So usually, the bad one, or the bacteria that are usually in human, although they are good or they are bad bacteria in the human, they are all heterotrophic. That's why they reside in the human body, because they can extract the organic carbon source from our body. So that makes them heterotrophic. Okay. The next few slides will show you on the types of growth. Okay. So the types of growth, it depends on the culture media, the platform, the medium that we use to grow them. Okay, so different types of bacteria, they need different types of growth requirement. So that's why in different culture media, they will have different nutrition to support the growth of these different microorganisms. Okay, so in this case, we are going to show you only 8 types of culture media, but in real life there are a lot of culture media. Okay, so the first one is the minimal media. Okay, so this is minimal media. So minimal media, from the name itself, is the bare minimum. So it's very simple, and because it is very simple, it is not usually used in the diagnostic clinical microbiology. So if you want to use it only in our lab, just to play around, just to practice with on how to culture the bacteria, you can use the minimal medium. But if you are in the professional lab certified, microbiology lab certified, where that lab is used to do medical diagnostic, then minimal medium is totally out of the place. Okay, so they can't use the minimal medium because it will give us a lot of data, which most of them are useless. The second one is called the nutrient medium. So besides minimal medium, the nutrient medium is the most common medium as well to use. So usually in our lab, we will use the nutrient medium. I don't think we have the minimal medium in our lab. Okay, so it is made of extracts of meat or soybeans, and basically there are quite the same as the minimal medium, but it is not as simple as the minimal medium. The third type of culture media is the enriched medium. Okay, so from the name itself, enriched, it means that that medium is added with something else. Okay, so for example, it is added with extra growth factors to what? Okay, to encourage the growth of something. So in this case, extra growth factors such as blood. Okay, so this is the enriched medium. So that's why it's red in color, because it is added with blood. So basically it is a nutrient agar, okay, just like number 2, but then it is enriched. It is added with a 5 to 10% of blood to encourage the small numbers of organisms to flourish, because in the blood it has the nutrients needed to grow the microorganisms. Okay, the fourth one is broth. Okay, so broth is, you can easily identify them because broth, it is in liquid, and when it is in liquid, it has to be in the test tube like this. Number 1, 2, 3, the minimal, nutrient, and the enriched medium, they are all in the Petri dish. Okay, so it starts with the liquid, and then when we pour it into the Petri dish, and then it will solidify into solids. But number 4, broth, they are still in a liquid state, and therefore they have to be in the test tube. Okay. The next one is non-selective medium and also the selective medium. Okay. So from the name itself, you can easily understand what they mean. Okay. So from the non-selective medium, it means that they do not select anything. Okay. So they support the growth of most non-fastidious microbes. Okay. So non-fastidious meaning yang paling tak cerewet. Okay. So fastidious microbes ni, it means that a group of microbes that they are very cerewet, that when they need to grow, they need to have a specific nutrient. Okay, so if you have a fastidious microbe, you can't use the non-selective, because non-selective, it is for the not cerewet bacteria, because it does not select against anything. Now, in contrast to the sixth culture media, it is the selective medium. Okay, so from the name itself, it means that it selects only a type of bacteria to grow and do not allow another type of bacteria to grow. So what do we have in this selective media is we have some additives that will inhibit the growth of some bacteria while allowing others to grow. Okay, so this is very useful when you have in your mind that you only want one type of bacteria to grow. So for example, you have a bacterial culture with a lot of bacteria inside there, but you only want E. coli to grow only. Okay, so you can use this selective medium. So only the colony appears on that selective medium, that is the E. coli that you want. Other than that, they won't be able to grow. The seventh culture media is the differential medium. Okay, so there is there is a confusion between the selective and also this type of differential medium, because sometimes they think that, okay, what is the difference between the selective and also the differential medium? Selective and differential, somehow they do select certain bacteria only to grow, but for differential, it allows the visualization of the metabolic differences in bacteria. Okay, so we have to remember the different types of the culture media, that is based on the nutritional requirement. But for differential medium, besides the nutritional requirement, it also selects, it also differentiates the bacteria based on the differences of their metabolism. Okay, so if you want bacteria only that use carbon only, or use oxygen only, or use nitrogen only, those are all the metabolic processes, then you can use the differential medium. And the last one is the transport medium. So from the name itself, it is a medium that we use to carry, to transport our bacteria from one place to another place. Okay, or the other name is the holding medium. So we just want to hold the medium so that it is stable, but we do not want them to multiply, because we want their exact number from the time that we take the sample from point A and then to transport them to point B. We don't want them to multiply, because by the time we receive point B, where is the place for the analysis, we analyze the number of cells. We want to know what is the exact number of cells when we take the samples in point B. Okay, so for example, this one, is the viral transport media. So this is the for viral, okay? The one we're talking is for the bacteria, but it is same applicable to the virus as well. And this is what they use for COVID-19. Okay? So you have to just imagine, because a lot of swab tests for the COVID-19, they are being done in a lot of places. Okay, so for example, they do the swab test in the UTHM campus PAGOH. So swab test using the cotton swab, and then once they have the samples from us, then they will put it into this viral transport media. Okay, so this viral transport media, they will hold the samples, okay, in this vial, and they will transfer the medium, the vial, into the lab for analysis. Okay, so there, when they analyze our sample, then they know whether we have a virus cells in there, whether we are positive or negative for COVID-19. And that's why it's very important that transport medium does not allow multiplication. Okay, so this is a table showing the differences between the selective and also the differential media. Because as I mentioned just now, a lot of people are confused with the selective and the differential media. But by now, I hope that you understand the difference between the selective and differential media. Okay, so selective is mainly on the nutritional requirement. So it has additives as well. Differential has additives, but for selective, it has additives so that they can inhibit some growth of microorganisms while allowing a certain type of microorganism to grow. But differential, besides differentiate them based on the nutritional requirement, we are differentiating them based on the metabolic differences as well. Okay, so the selection of the primary culture media will vary from lab to lab. This is true because, for example, just now we have learned about the 8 types of the culture media. So how do we know in this lab what do you use as the primary culture media? Okay, so there are few factors. Okay, so for example, the first one, the main factor is on the budget. Okay, so different cultural media, they have different price to it. Okay, the most simple one, for example, the nutrient medium, the minimal medium, then they are, because they are the most simple one, then they are the cheapest one. Okay, so if the medium that they use or the medium that they have, it has more complex substance in it, then it will cost you a lot. The second factor is on the type of microbe. Okay, so in that lab, we have to know whether you are dealing with what type of microbes, whether they are the pathogenic and also the non-pathogenic. Okay, so if you are dealing with the pathogenic bacteria, then the culture media will be different. If you are dealing with the non-pathogenic bacteria, then it will be different. And the type of microbes that the lab is dealing with, that influence the third factor, which is the type of lab. So if the lab is dealing with pathogenic bacteria, then we have a level clearance, the clearance level for each lab. So not all lab are allowed to deal with the pathogenic bacteria. Yeah. So for example, our microbiology lab in UTHM campus, PAGOH, we do not have the clearance level. We do not have the permission to deal with the pathogenic bacteria. So only the non-pathogenic bacteria. So how to prepare the culture media? Okay, so from eight culture media that we just learned, each one of them has different instructions, has different guidelines on how to prepare the culture media. So basically, they are very simple. Most of them come from a bottle, okay, a powder, and then you just use the instruction label on the bottle. You can just use them, okay, or you can refer to a lot of websites in Google, okay, where they show you on how to prepare the culture media. Okay, so just refer to the books, refer to the websites, and the most reliable reference is the label on the box or the label on the bottle itself. How to select the right culture media? Okay, so we know that we have a lot of culture media, not just the 8 ones that we just learned in this lecture, but we have a lot. Okay, so what are the factors, what are the justifications that can assist us in selecting the right culture media? So there are few factors. Okay, so the first one is the starting material. Okay, I hope you can understand my handwriting. So the starting material, it means that whether you are given a bacterial culture where you have a lot of unknown bacteria in it, or you need to collect the sample itself. Okay, so whether you are given the bacterial culture, or we have to self-collect the bacteria. Okay, so bacterial culture, usually maybe you can just take it from your water sample, okay, from tasik, from sungai, from longkang. Okay, you can just take that, and in that bacterial culture, they must have a lot of bacteria inside there. Okay, it's just that we don't know what. Alright. The second one is the self-collect. So it means that you are not given the bacterial culture, but we have to self-collect in terms of we have to swab it. Okay. So for example, you can take the bacterial sample from your handphone, from the doorknob, from your face, from your cheek. Okay. So this is the first factor that you need to consider when you want to do the when you want to select the culture media, because it shows that it helps you in choosing whether you need to use the broth, or you need to use the solidified agar plate, and from that solidified agar plate, which culture media that you have to use. Which brings to the second factor, the objective of your project or the objective of your culture. So if you only want to grow, or maybe you want to identify, or maybe you want to isolate them. Okay, so these are the 3 objectives. When you have the bacteria culture, then it will determine on what is the suitable and what is the right culture media. Okay, so for example, if you just want to grow them, then you can use the most simple, yeah, the minimal or the nutrient agar. But if you want to identify and isolate, then you have to use all those selective or differential media. But you have to really know you want to identify or you want to isolate based on what nutritional requirement. Then it will lead you to the right culture media. Okay, and the third factor is the origin of bacterial sample. Okay, so origin. Okay, ni salah je. Origin of bacterial sample. Okay, so the importance of us knowing the origin of bacterial sample, because from there we can do a research, we can do a literature review on what are the common types of bacteria that are usually present in that bacterial sample. Okay, so for example, the origin is a water sample. You take it from the kolam, from the tasik, from the lopak air, okay, or it can be from the surface sample. So this one, just now like I mentioned, you can take it from your phone, you can take it from your doorknob, face, cheek, or your laptop, okay, any surface. And the last one is from the air sample. So air sample is, our surrounding. So it can be your breath as well, okay, daripada nafas kita. So this different origin of the bacterial sample, they tell us the different common types of bacteria in it. Okay, so if we know that the origin is from water, and then we go to the literature review and they say that, okay, water usually they have this type of microorganisms, and we know that this type of microorganisms, then we can use the right culture media. Okay, so when we want to grow them, of course, the growth of any living things, they are influenced by the environmental factors. So in this case, we're going to share with you the 3 topmost influential environmental factors in microbial growth. So the first one is the pH. Okay, so mesti penting, is very important. So you have to prepare your media, your culture media. The pH must be between 7.0 and 7.5. Yeah? So that is macam neutral nak ke alkaline sikit. So we know that most bacteria, they can live in this pH range. But we also need to understand some of the bacteria, they can live in the acidic, the most acidic, and also the most alkaline. So that is why it's very important for you to understand the bacterial sample that you have, or maybe if you already know the type of bacteria that you have, you just want to grow them, okay, nak banyakkan mereka, then you have to study what are their nutritional requirement. So the second one is the temperature. Okay, so most bacteria, they are used to the human body temperature. Okay, so usually in our lab, in the incubator, we will set the incubator between 35 to 37 degrees Celsius, because this is the normal human body temperature, and that is the normal and common types of bacteria that they grow in. However, we can't forget Another type of bacteria that live in the extreme environment. So in the extreme cold temperature, in the moderate temperature, and also in the high temperature. So again, we have to know what type of organism that we want and their requirement to grow. And the third one is the oxygen requirement. Okay, so there are 5 different categories of bacteria when we want to categorize them in terms of the oxygen requirement. Okay, so let's have a look at this diagram. So this arrow at the bottom of the diagram, it shows the percentage of oxygen. To the right is towards less oxygen, and to the left it is towards more oxygen. Okay, so let's see this obligate aerobes. So obligate aerobes, it falls in the more oxygen. So it means that the obligate aerobes microbes, they need a lot of oxygen. And this is in contrast to the obligate anaerobes. Okay, so remember, aerobes, it means it must have oxygen. Anaerobe, it means it doesn't need much oxygen, it doesn't need oxygen at all. Okay, microaerophiles. So microaerophiles, they are still in the category of needing the oxygen, but not as much as the obligate aerobes. Okay, so in contrast to the aerotolerant anaerobes. Okay, so it is in the category of not needing the oxygen, but they also need the oxygen, but it is in the small quantity. And the last one is this one in the middle, facultative. So it's basically facultative. The spelling this salah. Okay, so facultative anaerobes. So this category of microorganism, of bacteria, they are in the middle of the scale though. So it means that this type of bacteria, they can grow with or without oxygen. If there is an oxygen, they can grow. If there is no oxygen, they can also grow. Okay, so facultative anaerobes ni paling muntage lah, paling best lah. Okay, so if the bacteria can grow in, if the bacteria is categorized into the facultative anaerobes. Okay, so bacterial growth. So when they grow, besides all those nutritional requirement, all those environmental factors, we need to know how they reproduce, how they replicate. Okay, so undoubtedly bacteria, they reproduce through a method named binary fission. Okay, so binary fission, it is a process illustrated in this diagram. Okay, so this is the first parent cell, okay, with the bacterial DNA inside, dekat dalam sel tu. And then the DNA replicate. So dia bila replicate, maknanya it makes another copy of the DNA. And because of that, then they go into the process of cell elongation. So dia panjang sikit daripada parent cell. And then septum formation. Ni dia punya cleavage dia tu. Okay, because dia dah nak mula untuk separate, which goes to the cell separation, and each new cell will receive a new copy of the microorganism. So this is binary fission. And then each new cell, they will undergo another binary fission. This one pun undergo another binary fission. So that's why dia akan boleh banyak. So when we have successfully grown them, okay, nak buat banyak-banyak, then how to determine the number of cells that we have successfully grown, okay. So there are three ways, three methods on how to determine the numbers. So the first one is on the direct counting under microscope, okay. So it is as simple as you put the samples in the microscope slide, and then use the microscope to count the live cells and also to count the dead cells. Second method is through the direct plate count. So this is not using anything, but just use your naked eye and use the plate where you grow the bacteria. And the third one is by using a machine, and this is what we call the density measurement or the optical density, because it uses a light so that it can tells us the absorbance based on the turbidity, based on the density of the samples. Okay, so this is the first method, which is using the microscope. Okay, so kita letak the samples inside the microscopic slide, and then this is under the microscope. Okay, so kita count which one yang dead cells, which one yang live cells. Okay, but before that we have some treatment lah kepada bacterial sample tu. We put some chemicals, where yang chemicals yang dead tu dia akan dicolorkan, okay, so that we know under the microscope yang color ni, for example color purple, it means that it is dead. Kalau yang color not purple, then it is live. So this is the second method, which is the direct method using the plate, okay, plate count method. Okay, so this one we have to use the naked eye only. Okay, so we have four examples of the plate. Okay, so A plate, too much growth, hard to count the colonies. It's true kan? Banyak sangat ni, macam mana kita nak count berapa banyak kat sini kan? Okay, so plate B, the growth is not as much as the plate A, but it is still hard to count the colonies, sebab masih ada yang macam bergumpal kan. So it's very hard to count. Plate C, this is better, way better than plate A and plate B. But if you can see, some of the colonies dia macam dekat-dekat. Kan macam ni. Dia dekat-dekat. So how to know how many single colonies are there actually? Kan? So it's quite susah. Then plate D is the most ideal one. Okay, so kita tengok kita boleh kira je 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Then we can report ada 15 single colony in this plate. Okay, so that's why using a direct count plate ni, dia ada several factors lah. Okay, so if you have this plate A, then you kena streak balik plate tu, ambil lagi koloni, streak lagi banyak-banyak until jadi seperti plate D sahaja. Okay, so this is the third method, the density measurement, okay, using the OD, optical density. So it's using the light source, okay, light source here, and then this is our sample. Okay, so sample A, you can see that color dia tu is very terang, okay, dia clear, meaning that not many bacteria in here. So bila light masuk pun dia tak akan refracted. Okay, but in comparison to this sample B, dia kan ada banyak dot tu, dia turbidity dia tinggi, meaning that these samples have a lot of bacteria. Jadi bila light shine through the bacteria, jadi dia akan diffract. Okay, dia punya bakteria. And it will give us a number on the absorbance. So daripada situ kita boleh tahulah how many cells that we have successfully grown. Okay. So kita akan belajar sikit on the plasmid. Okay? Because bacteria is very useful when we want to study on the genetic elements and the genetic alteration and also the genetic mutation. Okay, because they are very small, because they are very easy to grow, and because their chromosome has been successfully sequenced, so we can know everything about the bacteria. Okay, so this is the normal bacteria that we're talking about. Okay, so this is the structure of a bacteria. So besides the cell wall, flagellum, cytoplasm, and the bacterial DNA, we also have this plasmid DNA. Okay, so plasmid DNA, it is actually an extra piece of DNA, extra chromosomal piece of DNA. So in the plasmid DNA, usually researchers will use the plasmid DNA for antibiotic resistance and other virulence factors. And when they want to so this plasmid DNA usually they are being passed from one bacteria to another bacteria. So that's why a lot of bacteria have that antibiotic resistance to a certain type of antibiotic, because the antibiotic resistance tu lies within this plasmid DNA. Okay, so jahatlah dia. Okay, so this is a video on the Plasmid 101. Later, I will give you the link to the YouTube video in Google Classroom. Then you can have a look. Okay, so the plasmid, they must replicate as well. Okay, because they need to make themselves a copy, kan? Because dia nak pas kepada bakteria yang lain. So they have 3 methods on how to replicate themselves. So the first one is this transformation. Okay, so transformation is as simple as bacterial cells, dia uptake another bacteria. Okay, dia incorporatekan kat dalam tu, and then kemudian through binary fission dia akan jadi another new cells. Okay, but the new DNA tu either dia incorporate into the DNA cell ataupun dia akan jadi separate plasmid. Okay, so ikut dia lah. The second one is transduction. So transduction is quite the same as the transformation, where it imports the DNA from outside, but it's using the bacteriophage. Dia akan inject viral DNA tu masuk dekat dalam bacterial cell. Conjugation. Okay, so conjugation ni dia memang lain, because conjugation ni on how they want to replicate is dia buat jambatan ni. Okay, so from the donor dia keluarkan jambatan, lepas tu dia cocok dekat recipient. So it serves as a bridge, as a link. Then any DNA punya material, plasmid apa semua, dia boleh travel through this peleus. Okay, through this bridge or link into the recipient. So nanti dia pun dapatlah a copy of plasmid. So just imagine if this donor it has antibiotic resistance kepada antibiotik, kan? Then bila dia transfer kepada recipient, then recipient ni daripada dia susceptible to the antibiotic, then they become the resistant to the antibiotic. Okay, so kalau this is a bacteria yang menyebabkan disease kepada human kan. Jadi dia akan macam menyusahkan human tu lah. Human tu asyik sakit je sebab doktor dah bagi antibiotik, tak jalan, because bacteria tu dia ada plasmid which contains the antibiotic resistance to that particular antibiotic. Okay, so this is the mutations. What is mutation? It is the changes that occur in the DNA code. So we know that DNA code tu dia ada sehingga ke transcription and the translation process. So translation process itulah yang mana dia ada menghasilkan protein. So mutation dia boleh kacau dekat transcription punya process phase, ataupun it can kacau in the translation punya process. So either way they will change the process. They will change on what protein the cell makes. Okay, so different proteins they will function differently. So dia akan jadi lain lah, dia jadi dah tak betul dah. Okay. So that's all for part 2, where we covered the microbial growth and also the nutritional requirements, okay, of the bacteria. So next part we will cover the remaining, which is the epidemiology and also the antimicrobial properties, okay, antibacterial properties. Okay, so that's all for today. Hope you enjoyed the lecture, and if you don't understand, you can ask me any questions. Okay, thank you.